All right. Hello, everyone. I am gonna go ahead and do the exam 3 practice exam. So let's just jump right into it. Okay. So question 1 is asking which of the following membrane proteins tend to lack hydrophobic regions on their surfaces. And then we have A, integral membrane proteins, B, transmembrane proteins, C, integrins, or D, peripheral membrane proteins. So I can't really recall if we learned about integrins, but integrins are basically just, I believe, proteins that are just integrated within the membrane. So let's go ahead and draw a phospholipid bilayer. Just going to shorthand it a little bit. So this is our phospholipid bilayer, right? We have the hydrophilic heads facing outwardly, and we have the hydrophilic heads facing outwardly, and then the hydrophobic tails facing inwardly. So this, like I said, this is just a shorthand. So normally your phospholipids will look like this, and then the lipid bilayer is going to look like this, right? So now when the question's asking which of the following membrane proteins tend to lack hydrophobic regions on their surfaces, well, let's see. If something lacks hydrophobic, if something lacks a hydrophobic region, well, then it's going to want to hang out on the outside of the cell membrane, right? It's not going to want to be on the inside because if it had hydrophilic regions on its surface, right, and it was on the inside, well, that wouldn't work because remember, as a principle, like dissolves like. And in this case, we're not really dissolving anything, but it's still important to note, right, that, or maybe even like mixes with like, because hydrophobic region is not going to mix with hydrophilic region. It's just not going to work, right? So, what does that mean? So it means that transmembrane proteins. So what is a transmembrane protein? It's a protein that goes through or across the membrane, which could be like a channel protein or a protein pump. So this would be a transmembrane protein or an integral membrane protein. So an integral membrane protein is just something that's like kind of hanging inside of the bilayer, right? Just like an integrin as we were discussed. So what's going on here? So on the outside, just like the lipid bilayer, these proteins are what is known as amphipathic. So what does amphipathic mean? Right? So amphipathic means that it's both hydrophobic and hydrophilic. So it's going to be just like the lipid bilayer. It's going to be hydrophilic on the outside, facing the outside, and it's going to be hydrophobic facing the inside because just like our bilayer, it's hydrophobic on the inside and hydrophilic on the outside. So what then? We only have peripheral membrane proteins left. So peripheral membrane proteins, just like the name indicates, are going to be on the periphery, aka the outside of the cell membrane, outside the cell membrane. So that's where your peripheral membrane proteins are going to be on the outside. So so if it's going to be on the outside, if it's hanging around if it's hanging around the phosphate groups on the phospholipid, which are polar, right? This one's going to lack hydrophobic regions on the surface because if it didn't lack hydrophobic regions on the surface, then it would be on the inside of the cell membrane. But since it is lacking it, it would be on the outside. Hence, the answer is D. So let's move on. So let me pull up question 2. Okay. So question 2 is asking How do unsaturated fatty acids help keep any membrane more fluid at lower temperature? So, A, A, the double bonds form kinks in the fatty acid tails that prevent adjacent lipids from packing tightly together. B, the double bonds result in shorter fatty acid tails and therefore thinner membranes. C, unsaturated fatty acids are more polar than saturated fatty acids. Or D, unsaturated fatty acids have a higher cholesterol content that prevents adjacent lipids from packing together. Okay, so just from this, we can debunk a few things. All right, so let's draw an unsaturated fatty acid, right? So let's just go ahead and draw a phospholipid, yes? And then we have that's the glycerol backbone and then the phosphate group. Okay? Whoops, okay, sorry. So once again, this is a bit of a shorthand. I'm sorry, I'm not very good at drawing this. Oh my, please bear with me one moment. Okay, I'm just gonna wing it. I guess, okay, yeah, good enough. Let's do another one. Okay, so remember, for a fat to be unsaturated, it needs to have double bonds, right? So these double bonds, for these double bonds to exist, this, especially more specifically, this fatty acid tail is now unsaturated. And this fatty acid tail is also unsaturated. So if these double bonds did not exist, then it would be saturated, right? So let's go one by one. All right. So answer B is asking the double bonds result in shorter fatty acid tails and therefore thinner membranes. That is untrue. So length and double bonds have nothing to do with each other. They are not related. So you could have a very, very long fatty acid with no double bonds. Or you could have a very, very short fatty acid with no double bonds. And same thing with double bonds. You could have a very, very long fatty acid with double bonds and vice versa. So that one's not true, right? Because for that to be true, they would have to be related, right? Which they're not, right? So how about unsaturated fatty acids are more polar than saturated fatty acids? Well, that one's also not true because the presence of double bonds has nothing to do with polarity, right? We're not talking about polarity. This is pretty much the exact same polarity as if it was saturated, right? So that doesn't help keeping the membrane more fluid, right? That's just completely untrue. Unsaturated fatty acids are just as nonpolar as saturated fatty acids. The only thing that makes something polar if it has a polar covalent bond, such as oxygen and hydrogen, or carbon and oxygen, that sort of thing, or nitrogen and hydrogen. So how about D? Unsaturated fatty acids have higher cholesterol content that prevents adjacent lipids from packing together. So this one's also not true because unsaturated fatty acids and cholesterol are two completely different things. Cholesterol, let me draw out cholesterol real quick. Cholesterol is its own molecule, right? So this is cholesterol pretty much. Just like I said, you're gonna have to bear with me. This is what cholesterol looks like. It has like a four-ring structure, and this is common with This is common with most steroids, right? So if this is cholesterol, does cholesterol look anything like this? No, right? Because they're different molecules, but they're both still lipids. But and it's also important to note that cholesterol still has an important role in maintaining fluidity within a cell membrane, but unsaturated fatty acids cannot contain cholesterol because they're two different molecules, right? So the answer cannot be D. So that leaves A, right? A says double bonds form kinks in the fatty acid tails that prevent adjacent lipids from packing together tightly. That's exactly right. right? So recall, right? Butter, something like butter is fat, right? That's pretty much entirely fat. But also, right? Butter is solid at room temperature. That's because butter contains fully saturated fatty acids versus something like olive oil, which contains double bonds and is unsaturated, right? So what does this mean? Well, butter still melts at relatively low temperatures, right? If you stick butter in a pan, it'll melt easily, right? But that's just what that just says, right? Is that because it's lacking double bonds, it's able to pack tightly together. It's able to hold itself at higher temperatures, right? So going the opposite way, if something's at lower temperatures, right? So butter would be solid at low temperatures, but oil still wouldn't be solid at low temperatures. So, like I said, going the opposite way, right? Going the opposite way, if we have double bonds and the double bonds prevent tightly packing together the tight packing of molecules, then adjacent lipids would not be able to pack tightly. So that would make the membrane more fluid at lower temperatures, right? So more double bonds, more fluid at lower temperatures. Double bonds, more fluid at high temperatures. Because if we had double bonds, if we had unsaturated fatty acids like this one at higher temperatures, then the cell would be too fluid. So when we're talking about double bonds, the presence of double bonds in fatty acids in the membrane, we're talking about its fluidity and how it maintains the fluidity. Because there are three things in a cell. There are three things in a cell that are very important for it to maintain, right? And that's fluidity, acidity, and temperature. These are your three things that are very important to maintain in the cell, right? Fluidity, acidity, temperature. And temperature affects fluidity, right? So, but fluidity is entirely dependent on how many double bonds or how much cholesterol is in the cell membrane. So, moving on. So, question 3. So, question 3 is asking what kind of molecules pass through a cell membrane most easily? A large hydrophobic molecule. B, something that's small and ionic. C, something that's large and hydrophilic. Or D, something that's small and hydrophobic. Or E, something that's large and polar. Okay, so once again let's look at our lipid bilayer. So our lipid bilayer, just like I mentioned before, is amphipathic. So that means that the polar heads are facing outwardly and the nonpolar tails are facing inwardly. So what does this mean? It means that anything that's charged or polar is not able to go through. Right? Especially charged. Charge is like a triple X. That will never ever go through. Very, very, or I don't want to say never ever, but it will almost never go through. Versus something that's nonpolar or small. These will go through. So, what is nonpolar and small? Oxygen, which looks like this, and then carbon dioxide, which looks like this. And I hear you saying, "Well, wouldn't CO2 be polar because it has carbon and oxygen, and wouldn't that make a polar bond?" Well, you would be right, but there's what's known as a dipole moment, and essentially, dipole moment is where the electrons are attracted to each other. Sorry, where the electrons are attracted to one specific part of the molecule. But if you would see here, carbon is attached equally to two oxygens, so these would cancel each other out, so making CO2 a nonpolar molecule. And one more thing. So I know I said polar molecules are not going to pass through the cell membrane easily. However, water is the only exception to this. Water is the magical exception to this. So of course water would be that, right? So these are your only exceptions. Something that's nonpolar and small is just able to go right on through. Check. So, going back, right? Something that's charged, never gonna go through. So that includes like ions. So that includes like something like sodium, right? Or potassium. These are never going to go through. They're just gonna be blocked off by this non-polar region in the middle. And same thing with polar molecules. So if we have something like sugar, for example, so let's draw a sugar. So sugar has a bunch of OHs on it, right? A bunch of OHs. That's like the key identifier of sugar. So what are OHs? Those are of course polar. So because it has so many of these polar groups, it is not able to pass through easily. Right? So what does that leave with us? That leaves us with the size of molecules, right? So if something is large versus something is small, something that's large. So look at how tiny these gaps are, right? If you have something that's large, like for example, I don't know. Let's draw like a quick. So this would be an example of an alkaloid, which you don't know what alkaloids are, but that's okay. So this is large. This would be considered large and nonpolar. This is not going to be able to pass through easily. That isn't to say that it won't pass through at all, but because it's still nonpolar, this molecule is still nonpolar. It's going to pass through, but not as efficiently, right? It's not going to be able to pass through very effectively because it's so large, because we're fitting such a large molecule into such a small gap. Right, so what does that leave us with? So large and hydrophobic, that's wrong. Small and hydrophobic, oh, that one's right, but we'll get back to that. Small and ionic, well, when it comes to ionic, as soon as something's ionic, that's how you know. It's just not going to go through. So small and ionic is also wrong. Large and hydrophilic is also wrong. And then large and polar is also wrong. So the most easy thing that's going to pass through the cell membrane is small and hydrophobic, as we discussed here, right? But it's important to note most easily. So like I said, large and hydrophobic is still going to go through a little bit, but what's going to go through most easily? Small and hydrophobic. Let's move on. Okay, so question 4. Which of the following is a characteristic feature of a carrier protein in a plasma membrane? So, A, it works against the concentration gradient. B, it is a peripheral membrane protein that does not embed in the membrane. C, it is specific for a particular type of molecule. Or D, it requires the use of energy to function. Okay, so if you'll give me 1 second. Okay, so sorry about that. I just needed a quick reminder of what a carrier protein was. So, as I thought, right, a carrier protein is once again embedded in the cell membrane. So let's draw our cell membrane once again. We're going to be seeing that a lot. So, anyway. Actually, it would be easier if I just showed you a picture. So let's do that instead. Wow, that's not grainy at all. Anyway, MS Paint, am I right? Ha ha ha. Anyway, so when we're talking about carrier proteins, what they do is recall back to the previous question, right? We can't carry ions, and we can't carry something like glucose, which is large and polar. Those can't go right through the cell membrane. That's just not going to happen, right? So what it needs is the help of a carrier protein. So now what a carrier protein does is that it has a specific spot, like right here, for example. Let me draw this in yellow. Right here, it has a specific spot for something like sodium, or for something like glucose, or for something like potassium, as another example. It has a specific spot where it could fit in, right? It could it can go inside the protein, and it can just go right out into the cell membrane. So it's important to note that this is a carrier protein, not quite a pump. A pump would be different, right? So there's a difference between the carrier protein and the pump. What the pump does, the pump is a form of the pump is active transport, whereas this is facilitated diffusion. So active transport versus facilitated diffusion, one works against the concentration gradient, one works with the concentration gradient. This one is working with a concentration gradient. So as you can see, we have a high concentration of particles on the outside of the cell membrane, and then we have a low concentration of particles inside the cell membrane. So look at where the particles are moving. It's moving with the gradient. So it's working thermodynamically favorably. So this does not require energy. So these are carrier proteins. They are literally carrying particles from one side of the cell to the other. So let's look at our answer bank, right? Works against the concentration gradient. That's wrong because that would be a pump. Is the peripheral membrane protein does not embed in the membrane. Well, that's also wrong because it would have to go through it would have to go through the cell in order to be transported. So it has to embed with the membrane, and then requires energy to function. Once again, that would be a pump. So that leaves with answer C. It is specific for a particular type of molecule, which is what we discussed, right? It has a spot for a molecule. So this is kind of oval shaped, right? If we had something that was, for example, hexagon shaped like sugar, it would not be able to go through because it's not shaped like that. So moving on. Okay. Which of the following molecules would you expect to diffuse to the lipid bilayer of plasma membrane most slowly, if at all? Okay, so let's just go over this. This one's real quick. This one's like kind of review of the previous question. Okay, so let's look at this. Oxygen, that's going to go right through. That's both small and nonpolar. Methane, small and nonpolar, once again. Water is the exception. CO2, small and nonpolar, leaving a sodium ion as the correct answer. Now remember, ions are basically never going to go through. Almost never going to go through. It's very, very, very unlikely for a sodium ion to cross the lipid bilayer. But methane, which is a hydrocarbon, oxygen, which is nonpolar, water, which is our exception, and then carbon dioxide, which is also nonpolar. All of those are going to go through willy nilly. They're going to go through really easily. So, moving on, question 6. I feel like I get a lot of questions about tonicity and whatnot, so let's go over this one kind of slowly. So, the question is asking, mammalian blood contains the equivalent of 0.15 molar sodium chloride. Seawater contains the equivalent of 0.45 molar sodium chloride. What would happen if the red blood cells are transferred to the seawater? So, answer A is saying water will leave the cells, causing them to shrivel and collapse. The blood cells will take up water. B says the blood will take up water, swell, and eventually burst. C, salt will be exported from the blood cells and by facilitated diffusion. Or D, NaCl will passively diffuse into the blood cells. Okay, well, off the bat, we can eliminate D because nothing. This is an ionic compound. This will form sodium and chlorine ions in solution. So in no like going back to the previous example, the previous question, in no world will this passively diffuse into red blood cells. So that's out of the question. And we can eliminate C because NaCl will be exported from the blood cells by facilitated diffusion. That's also out of the question. Which, I mean, yes, that might happen. And sorry about that. Yes, that might happen in certain cases, right? I mean, facilitated diffusion. I mean, sure, I guess that might happen. But in this case, we're talking about osmosis. So now, osmosis, we're talking about diffusion. Diffusion is the movement of particles from high to low. Osmosis is the movement of water, and specifically water. Only water can do osmosis. So water is the special exception. So when we're talking about the movement of particles always goes from high to low, right? That's the concentration gradient. But if we're talking about water, then we go from low to high. So what does this mean, right? So water always wants to balance out. Water always wants to be, it wants to balance out, for example, in this case, 15 molar with 45 molar. So the more solute you have, by definition, the less solvent you're going to have. Right? So if we're low in solvent, then we're going to want to move to areas of higher solvent. So that's the movement of water versus the movement of particles, which is from high to low. So movement of water is always from low to high. So what does this mean then? So low to high, hypo to hyper tonic. Right? That's what that means. High movement of water from low to high, movement of water always goes from hypotonic to hypertonic. And I also want to emphasize, hypo and hypertonic need to exist with each other. They exist with context. They exist together always. Right? So nothing can be low if it doesn't have something high to be compared to. So in this case, right, something with 45 molar versus something with 15 molar. Molar is just a measure of concentration of solute, right? Molar is equal to mole, if you're taking chemistry, moles over liter, which you don't really need to know that. You just need to know higher number, we're going to be more concentrated. Anyway, in this case, the seawater with the equivalent of 0.45 molar is going to be hypertonic versus the 0.15 molar, which is now going to be hypotonic. Now, if the seawater had an equivalent of 0.1 NaCl, then the seawater would be hypotonic and the blood would be hyper. But since that's not the case, anyway. But I just want to emphasize, right? It's always going to be relative. So in this case, the seawater is going to be hyper relative to hypo. So let's draw a little container, right? Let's make the water blue, of course. Let's make the red blood cell red, obviously. Okay, So now our red blood cell contains 0.15 molar. And then our water, the water on the outside, contains 0.45. So 0.45, that's of course going to be hyper, and 0.15, that's going to be hypo. Right? So, what does this mean? So we're always moving from hypo to hyper. So the water on the inside of the cell is going to move outwardly. So now what does this do? Now, since the water is leaving the cell is going to shrivel and collapse. It is not going to have the water in it to sustain itself. So that leaves answer A. Water will leave the cell and cause it to shrivel and collapse. Now, if it was the opposite case, if the blood cell was hypertonic, then water would move inside, causing the cell to burst. So that's all tonicity. That's all like concentration, movement of water, osmosis. So yeah, that was your review over tonicity. And actually, one more thing. So let's say both the blood cell and the water. But the blood cell and the water had a concentration of 0.15 molar. Now these are going to be isotonic. Now what does isotonic mean? So iso means same. You think of isosceles triangle, or that's the only example I could think of. It means same or means equal. So the movement of water from the inside of the cell is going to be the same of the movement of to the outside of the cell. These are going to be equal. Now that isn't, like I said, that isn't to say. There's still movement of water, right? The water is still moving because everything is constantly moving, right? The water is still moving inside and outside of the cell, but the thing is, it's moving at an equal rate. Right? It's moving at an equal rate. So now water is going to go inside at the same rate as it's going outside, hence isotonic. So there was your review over tonicity. So let's move on to question 7, which just looking at the time, it's been already half an hour, and I have quite a bit of exam left, so I'm just going to kind of speed this up a little bit, hopefully. So question 7. So the question is asking, which of the following membrane activities requires energy from ATP hydrolysis? So A, the movement of glucose into a bacteria cell medium containing a higher concentration of glucose than inside of the cell. B, movement of sodium ions from a lower concentration in a mammalian cell to a higher concentration in extracellular fluid. C, movement of carbon dioxide outside of the cell. D, facilitated diffusion of chloride ions across the membrane through a chloride channel, or E, movement of water into a cell by osmosis. So this question is asking about active versus passive transport. So the difference between those, right? So we have active versus passive. So for passive, there's no for passive, there's no energy input. And for active, there's energy involved. So now what does this mean? It essentially means that if there's no energy involved, then it's thermodynamically favorable and it's going to happen naturally. If it's active, then and there is energy involved, then it's not thermodynamically favorable and it needs the assistance of something that contains a lot of energy, but a lot of potential energy, right? So if there's energy involved, then we have to do something in order for in order for transport to happen. So that's the difference between active and passive. So movement of water into a cell by osmosis, that's going to be passive, right? Facilitate diffusion immediately, that's passive. Movement of carbon dioxide outside of a bacterial cell, just like movement of water, that's going to be passive, that's going to be simple diffusion. And movement of glucose from higher to lower, that's passive. Leaving only movement of sodium ions from a lower concentration in a mammalian cell to a higher concentration. So going back to just removing the process of elimination aspect of it, we know this because which of the following membrane activities requires energy from ATP hydrolysis? So when we're hydrolyzing ATP into ADP, right? When we're doing this, we're releasing energy, just like what the question says. It requires energy from ATP hydrolysis. So when we're requiring energy, then active transport's involved, which an example of that would be from moving from higher, from moving from lower to higher, versus passive, which is moving from higher to lower. Right. So I also want to write that down. So no energy input would be from high to low. And then energy input would be from low to high. Okay, so that was question 7. Let's move on. Okay, so sorry about this. Okay. Okay, this doesn't want to cooperate with me. Okay, I guess not. Never mind. Okay, so number 8 is asking proteins that allow the diffusion of ions across membranes in the direction of their concentration gradients are most likely A. aquaporins, B. carrier proteins, C. active transport pumps, or D. channel proteins. So, proteins that allow the diffusion of ions across membranes. So we can eliminate aquaporins, right? Because that only has to do with water. We're talking about ions in this example, so we can eliminate aquaporins. We can also eliminate active transport pumps because we're talking about the diffusion of ions in the direction of the concentration gradient. Active transport would be against. That leaves carrier proteins and channel proteins. So it wouldn't be channel proteins because the difference between it wouldn't be carrier proteins because the difference between carrier versus channel, it's kind of confusing because both of these work with the concentration gradient, right? So then it starts to get confusing, right? So what does this mean? So that essentially means that carrier proteins don't necessarily act by diffusion, right? They simply act by literally. Let's go back to the example, right? This is a good one. Like this is a carrier protein, right? It literally just acts by not necessarily by diffusion, but by just transporting one molecule to the other side, right? In that sense, it's not necessarily active, but there's still like some sort of activity going on. But because since we're still moving from high to low, um, it's still going to be passive. No, it's still going to be passive diffusion versus a channel protein. Which let me pull up a picture of a channel protein. So this is a channel protein. Channel protein. It's this. No, this one instead of just transporting it, right? It now acts by diffusion. Now we have a high concentration to a low concentration. That's what a channel protein does. It essentially just acts as a channel for something like ions or sugars or whatnot to just go right through the cell membrane. So that's a channel. So the answer is channel proteins, D. So let's go to question C. Sorry, question 9. Okay, so before we go into the next few questions, I think it would be beneficial for everyone if I just go over the cell organelles. So let's go over our cell organelles. So cell organelle review. So let's just list off the cell organelles as they come. Yes? Okay, so we have the nucleus, of course. Sorry, how do you spell nucleus? I think it's how you spell it. I'm gonna seem really stupid if that's not how you spell it. I already seem kind of stupid. All right, nucleus. Yep, that's how you spell it. Sorry about that. Okay, so nucleus, smooth ER, smooth endoplasmic reticulum. I think that's how you spell that. Rough endoplasmic reticulum. Let's just do this. Ribosomes, lysosomes, peroxisomes, cell membrane, slash. What's another name for the cell membrane? Anyway, cytosol, cytoskeleton, Golgi apparatus, mitochondria, I think that's most of them. There's probably one important one that I'm missing. Anyway, so everything that you see here are both in plant and animal cells. So let's plant exclusives. Cell wall, central vacuole, and chloroplast. Now the cell wall also exists in bacteria. But yeah. But these are your organelles. So, what do these do? And what do these look like? So let's look up the anatomy of a cell real quick. So then we could have a better idea of what we're looking at here. Because if I drew out the cell for you, then I would probably get fired, honestly, because the drawing would be so bad it would be offensive to your eyes. Anyway, so okay, well, I guess another organelle would be would be a vesicle, which it's not really an organelle because a vesicle is temporary and it's only used once, but some people still consider it to be an organelle. Anyway, so this is your cell, right? So what do we notice about the cell? So the cell has a bunch of different organelles, obviously, but we have a few different things going on here. So of course we have the okay. Let me do how about red? Oh, let's do yellow actually. No, just trying to see what color would actually work the best here. Let's do bright green. How about that? That's fun. Okay. So we have the nucleus, right? So now the nucleus is where we hold all the DNA, right? That that should be clear, right? So what does the DNA do? The DNA is like the center for instructions. Okay, rather the DNA is the instructions for life. There we go. So the DNA contains the instructions for creating proteins, and then those proteins can make everything else. So The DNA hosts all the information. It is the library of cookbooks, as Dr. Howe might put it. So connected to the nucleus, which is where we keep our DNA, is the rough ER. So what does the rough ER do? The rough ER is where proteins are made. So proteins are made here. And then after they're made here, then they turn into vesicles. Or the proteins that are made don't turn into vesicles, they get put into vesicles. So after they're put into vesicles, they go to the Golgi apparatus. So now the Golgi apparatus, what it's going to do is it's going to process the proteins slowly. Maybe not slowly, but it's going to process the proteins and it's going to create a vesicle. So we have the Golgi vesicle here, right? And then the Golgi vesicle is going to go out of the cell into where it needs to go, right? The Golgi apparatus just it's like a postal service, right? You put like the label on the box, and then you ship out the box to wherever it needs to go, right? That's what the Golgi apparatus does. So, what else do we have here? We have also the smooth ER. So the rough ER are what make the proteins, right? Now the smooth ER, the only difference between the rough ER and the smooth ER are the presence of ribosomes, right? So the ribosomes are actually what make the proteins themselves. Now DNA from the nucleus gets transcripted, and then it goes out of the cell into the ribosomes to get translated into a protein. So what you'll learn about more in the future, right? But for now, just know that ribosomes are what's actually making the proteins. And you have a bunch of ribosomes on the rough ER, which are going to go ahead and package the protein and send it to the Golgi apparatus, which is going to package that further and send it to where it needs to go. Versus the smooth ER. Now the smooth ER are where lipids are made. So now the smooth ER you would expect to find. Sorry. Now the smooth ER is where you would expect to find like where you expect like the very sorry, I'm tripping over my own words here. This it's just where you would find the center for lipid production. So what are lipids? Those are like lipid hormones such as sorry, having a total brain fart here. Lipid hormones such as testosterone and estrogen, as well as making cholesterol. So the smooth ER is just where we're going to make lipids, versus the rough ER, which we're making proteins. And the only difference is the presence of ribosomes. So we went over ribosomes, right? So how about lysosomes? Now lysosomes are basically what just break down. So lysosomes break down stuff that the cell doesn't need. So now lysosomes also take part in something known as autophagy. So what does autophagy mean? Auto means self and phagy means to eat. So literally lysosomes participate in self-eating, right? So what does this mean? It means that it's going to literally take dead organelles and useless things in the cell, and it's going to eat it. And that's what autophagy is. It's eating the cell. Sorry, the cell is eating itself through the lysosomes. So now the lysosomes have digestive enzymes in them that operate at high pH. Operate at low pH. And they're essentially like the cell's recycling center, as how Dr. Howe might put it. Because you put something in, and then something else comes out of it that's more useful to the cell. That's what the lysosome does. So how about peroxisomes? I don't think peroxisomes are labeled here. Oh wait, no, here we go. So here we have the peroxisomes. Peroxisomes are just like lysosomes, but they're more specific. Peroxisomes break down peroxides. That's really all you need to know. And it's not really something you need to really focus on. Peroxisomes break down peroxides. Peroxides are dangerous to the cell because they can cause genetic mutations. Anyway, cell membrane keeps everything inside, right? It mediates what goes in and what comes out. So that's what the cell membrane does. It also has a bunch of other functions, such as maintaining cell structure, maintaining the shape of the cell, also has to do with fluidity, also much of integrated proteins and peripheral proteins, and the cell membrane just has a bunch of stuff going on, which we've gone over previously with the proteins and whatnot. So then we have the cytosol. So the cytosol is just the liquid, or rather the fluid that everything in the cell is suspended in. So cytosol can also be considered like the matrix. Not like the movie The Matrix, but like, something that each organelle is suspended in. Like the cytosol is the matrix of the cell. And then we have the cytoskeleton. Cytoskeleton is just for transport, like within the cell. It's also primarily like the skeleton in a human is for maintaining structure in the cell, maintaining rigidity and whatnot. That's the cytoskeleton. It also has to do with the centrosomes. Now, the centrosomes are where the mitotic spindles come into play, and the mitotic spindles are made out of microtubules, which are a type of cytoskeleton, which I'm not sure if you need to know that. But Golgi apparatus, we talked about Golgi apparatus. And how about the mitochondria? Now, the mitochondria is one of those things where you joke about after you finish high school. It's like, oh, well, I came out of high school only knowing that the mitochondria is the powerhouse of the cell. What does that even mean? What does the powerhouse of the cell even mean? Basically, what it means is that the mitochondria is going to produce all of the energy that the cell needs. Now, that's a pretty tough job for the mitochondria, right? But it's very, very, very necessary. All the energy, all the glucose that we eat and consume, or fats that we consume, is processed here in the mitochondria, where it is then broken down into ATP. And ATP is the primary energy molecule of the cell. So that's what mitochondria does. It makes ATP. It is not the powerhouse of the cell. I mean, it is the powerhouse of the cell, but I want you to go beyond that. It goes beyond just being the powerhouse of the cell. It is the money maker. It is like nothing can proceed without energy. And that's what the mitochondria does. Other unique things about the mitochondria is that it, just like the nucleus, it has a double membrane. So let's write that down. So mitochondria has a double membrane. It has its own DNA. It has mitochondria DNA. So mDNA. Sorry. It has its own ribosomes too. So there's a theory that mitochondria were actually once prokaryotic cells, and then the earliest eukaryotic cells actually absorbed mitochondria, and it just worked out for both of them. So now mitochondria now exists within the cells, right? Because the mitochondria DNA is much more similar to that of a prokaryote. But that goes beyond what we're actually learning about. So anyway, what's not pictured here, because this is of course an animal cell, are plant exclusives. So like the cell wall and the central vacuole and the chloroplast. So the chloroplasts, they kind of work inversely in the mitochondria. Now the mitochondria works to break down sugars and to form energy. The chloroplast absorbs energy through sunlight, also through ATP. It absorbs energy to make sugars. That's what the chloroplast does. Which, this isn't really a plant biology class, so you really need to know that, but that's what a chloroplast does, and we're actually going to go over photosynthesis later this semester, I believe, but it's not a super big emphasis for this exam. Anyway, central vacuole, there's a vacuole pictured here, but in a central vacuole, you have the plant cell, and you have this big old thing in the middle. That's the central vacuole. So what the central vacuole does, essentially it just holds water, or toxins, or pigments. That's what the central vacuole does. So if you have like a rose, like the rose petals, for example, within the central vacuole, it would contain all the pigments for it to be red, or for it to be pink, or yellow, or whatever color the rose is. So that's what the central vacuole will do. It holds water, right? And it holds pigments, and it holds toxins. Now the cell wall behaves much like the cell membrane, except that cell wall, its primary purpose, unlike the cell membrane, which mediates what goes in and out, cell wall is just basically it just basically maintains the structure and rigidity of the plant cell or bacteria cell. So that was your cell organelle review. Now let's move on back to the, or let's do a rapid fire of the next few questions of the practice exam. Like I said, this is going to be a rapid fire, so I'm just going to include all of the questions. Whoops. Okay, so that's quite a lot of text, which, yeah, but anyway. So, one sec, let me decrease the size of the brush again. Let's make it a little bigger. That's good. Okay, so a bacterium engulfed by a white blood cell through phagocytosis will be digested by enzymes contained in the lysosomes. There are no digestive enzymes in vacuoles or secretory vesicles or peroxisomes. Well, yes, peroxisomes, but peroxisomes are only for peroxides, and there are no peroxides mentioned in this question, so it wouldn't be peroxisomes, nor in Golgi vesicles. So leaving the only answer, lysosomes, which, like I said, lysosomes contain digestive enzymes. So the answer is D for number 9. So just a reminder, the 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 prefix phag, right, means to eat. Cyto means cell. So phagocytosis means eating cell. So that's what's happening here. A bacterium was engulfed by a white blood cell through phagocytosis. The white cell is literally eating something, phagocytosis, and now it's going to be digested through lysosomes. Anyway, moving on. Number 10. Which of the following molecules or structures will be found in eukaryotic cells but not in bacteria? Well, a flagella will be found in bacteria. An ER will not be found in bacteria. And a plasma membrane, of course, will be found in bacteria. Oh, and then ribosomes and DNA. So the thing about bacteria is that bacteria have very few cell organelles. So what's going to be in a bacteria? Obviously, a plasma membrane. Otherwise, everything's going to just spill out. Also, DNA. All living things have DNA. Ribosomes. I mean, something needs to make the proteins. So bacteria also contain ribosomes. And then flagella. Flagella are present primarily in bacteria, but also in some eukaryotic cells, such as sperm cells. Anyway, ER. Bacteria do not contain membrane-bound organelles, and that includes a nucleus. Bacteria do not, once again, bacteria do not have membrane-bound organelles, leaving the only answer B, endoplasmic reticulum, which is a membrane-bound organelle. So, how about number 11? Which of the following structures will be found in nearly all eukaryotic cells? So this is a which of the following question. So I think there are multiple answers to this one. Oh wait, no, never mind. I should have read the question. Okay. So, which of the following would be found in nearly all eukaryotic cells? Not a flagella. Your average eukaryotic cell is not going to have a flagella, despite the fact that some do, but we're talking about nearly all. Nearly all eukaryotic cells will not have a cell wall. Only plant cells will have a cell wall, not animal cells. And same thing with a chloroplast chloroplasts are only in plant cells and some cyanobacteria, and a nucleoid that's only in bacteria cells, leaving mitochondria, right? And another thing that Dr. Howe wants to mention, or wants to make note of, is that mitochondria are in both plant cells and animal cells. It's not like plant cells go through cell respiration and animal cells go through cell respiration and plant cells don't. It's that both do. Both go through cell respiration because both have mitochondria. It's not like one has a mitochondria and the other has a chloroplast. No. one has both chloroplast and mitochondria, being a plant cell, and then one just has mitochondria, being animal cells. Anyway, large number of ribosomes are present in cells that specialize in producing which of the following? So remember, ribosomes, we make proteins via ribosomes. This is what my 7th grade biology teacher taught me. Ribosomes, think of ribs. Ribs contain protein because they're meat, right? Ribosomes, rib, protein, that sort of thing. So we could just skip right to proteins because they're not going to make lipids, they're not going to make nucleic acids. They're going to make proteins, and that's it. So which structure is the site of synthesis of proteins destined for export from the cell? Now here, we're still talking about the synthesis of proteins. So that wouldn't be free cytoplasmic ribosomes because that would be for proteins within the cell. When we're talking about proteins that are destined for export, then we're talking about the rough ER because we're still making proteins, but the proteins are actually going to go through the ER and through the Golgi and then outside. And that's part of what's known as the endomembrane system, which is we can go over after these. So number 14, which organelle or structure is primarily involved in the synthesis of oils? Smooth ER. Once again, we went over that. So we have the rough ER and the smooth ER. Smooth ER makes lipids. Rough ER makes proteins. And then we have number 15, which of the following statements correctly describes the function of the Golgi apparatus? Detoxification of drugs. That one's not it. Synthesis of cytoplasmic proteins. That's also not it. Because remember, Golgi apparatus do not do the synthesizing. Protein modification and sorting. That one's correct. And production of ribosomal subunits. That one is also incorrect. So, that would leave C. Protein modification and sorting. Once again, part of the endomembrane system. So what is the endomembrane system? We can go over that real quick. So endomembrane system. So we have the rough ER, and then vesicle from the rough ER. Then we do the Golgi, and then Golgi, vesicle, and then outside of the cell. And not included here. Well, it's still part of the endomembrane system, but we often forget about it. Or lysosomes, which break down waste from the endomembrane system. And there's a picture of it. Kind of. Well, let me pull up another one. So this is basically a picture of the endomembrane system, right? So we have the nucleus. So we have the nucleus, right? And the nucleus is you have all the instructions. So the instructions are gonna essentially be transported to these ribosomes, where it's gonna make a vesicle. Right? So you have the vesicles from the rough ER, which are gonna go through the Golgi to be processed. And then so now we have the vesicle from the Golgi that's going to go outside of the cell. Now this is your endomembrane system. Is that clear? All right, let's move on. I'm speaking rhetorically, of course. And there's a couple of other questions that I haven't included here, so let's go ahead and go on to those. Okay. Okay, so number 16. Tay-Sachs disease is a genetic abnormality that results in cells accumulating large, complex, undigested lipids. Which cellular organelle is likely to be defective in this condition? So once again, lysosome is our recycling center. If we're not properly getting rid of all the stuff in the cell, if we're not properly getting rid of all the waste material in the cell, then it's going to accumulate. And what breaks down the garbage of the cell? Lysosome. So it's going to be A as the answer. So now about 17. Which plant cell organelle contains its own DNA and ribosomes? So, so this is a bit of a trick question because you think plant cell organelle. Okay, well you immediately think of organelles that are exclusive to the plant cell. Well, look at these. The only thing here that's going to contain its own DNA and ribosomes are the mitochondria. Rough is not going to contain its own DNA. Vacuole is not going to contain its own DNA. Peroxisome, Golgi, same story. Like I said, this question is designed to trip you up. Don't be tripped up. Mitochondria exists in both plants and animal cells. So now number 18. Which of the following is a correct match between cytoskeletal elements and its functions? Now, I'm not exactly sure if you're going to be tested on this, but it is important to note, right? When we're talking about cytoskeletal elements and their functions, remember that recall that we talked about a little bit of microtubules and how they create the mitotic spindles during cell division. Excuse me, that's B. Now, microfilaments and flagellar motility. Flagellar motility, I think, are primarily made of microtubules. And then microtubules and cell separation. I think that one would be intermediate filaments. I could be wrong. And then microfilaments and intercellular vesicle transport, that would be intermediate filaments. So the answer would be B. But once again, I don't think there are any cytoskeletal elements that you need to be tested on. I could be wrong about that, and if I am wrong about that, then I will include it in the addendum after this stream is ended. If not, then you won't hear anything. Anyway, now why? So number 19 is asking, why isn't the mitochondrion classified as part of the endomembrane system? So recall we just went over the endomembrane system. That includes the lysosome, the lysosome, the rough ER, the Golgi, and then vesicles. So it would not be. So it's not involved in protein synthesis. It has a double membrane structure, but that doesn't have anything to do with it. And it is not derived from the endoplasmic reticulum or Golgi. That's also correct. And produces a special class of transport vesicles. That's also there are no transport vesicles from the mitochondrion. And I need a little drink of water for a sec. One sec. Excuse me. So, okay, so let's look at our answers again. So it's not involved in protein synthesis. It's not involved in protein synthesis directly, right? I mean, sure, it's not necessarily producing the proteins, but it produces the ATP for protein synthesis. That's necessary. So that one was going to be wrong, actually. So now it's not going to be derived from the endoplasmic reticulum or Golgi. Those are our elements of the endomembrane system. So the answer is going to be B. So moving on, there's a few more cell membrane questions that we can go over. Actually, there's just one more actually, and then we can go, and then we're gonna move on to. Sorry, now we're gonna move on to. Oh my god, I can't focus. I'm so sorry. Then we're gonna move on to cell division. There we go. Oh my god. Okay, so a vesicle transporting material between the rough ER and Golgi is called a transport vesicle. I believe. Yes. So phagosome, I don't think we learned about phagosomes, and then lysosome, that's not a vesicle. So that leaves transport vesicle and then secretory vesicle. So when we're transporting between the rough ER and the Golgi, that's good. We're not secreting anything out of the cell just yet. When we're transporting from the Golgi to the outside of the cell, then it would be a secretory vesicle because then we're actually secreting something out of the cell. Versus the rough ER and the Golgi, that is going to be a transport vesicle. We're moving the rough ER to the Golgi. Sorry, we're moving the vesicle from the rough ER to the Golgi. That would be transport vesicle. So, those are your cell organelles. I am getting a little tired. So, what I'm going to do is I'm going to do kind of what I did with the organelles, and I'm going to go over the elements of cell division. So I've been getting a lot of questions about this. So please, 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 I implore you to pay attention to this because, like I said, I've been getting this has been the most confusing topic from every student I've talked to. So let's talk about mitosis. Let's talk about the cell cycle, mitosis, and meiosis. So let's start with the cell cycle. So the cell cycle is, of course, a cycle, right? So we start with G1, and then we go into the S phase, and we go into G2, into the M phase, back to G1. So now the M phase takes up about like 10% of the time a cell takes up about 10% of a cell's lifetime. And you have this entire portion from G1 to G2 known as interphase. So we have interphase, and then we have the mitotic phase. Now the mitotic phase is kind of what we care about a little bit, because the mitotic phase, that's when the cell actually grows. Mitosis is cell growth, and of course cell division. And yeah, I don't have much else. So that's all happening during the M phase. Now during interphase is essentially just the cell preparing for mitosis, because that's a cell's primary function, is to prepare for it's just to make more of itself, right? But how does it do that? So during a cell's lifetime, it just exists during G1, and then it just exists during G2. That was a bad explanation. Sorry. So G1 is basically just like the ordinary cell function. Everything's normal. Everything's going into plan. All the proteins are being synthesized. All the DNA is doing its thing. But then the cell gets a signal here saying that, okay, well, we have enough energy and we have enough resources to synthesize DNA. So then that's what it's going to do. So during the S phase, that's when we're duplicating. That's when we're duplicating the DNA. And when I say duplicating, I mean duplicating. The amount of chromosomes does not change, but the chromosomes are still doubled. They create exact copies of each other, known as sister chromatids, which we will go over later. So after S phase, right? After everything's been said and done, we go into G2 phase, where now in G2 phase we still have sister chromatids, and now the all the DNA has been duplicated. So that's during G2. So now during G2, we have a few more checks here, right? Now what these checks essentially do is that it says like, okay, we have enough resources to do cell division, we have enough energy to do cell division, and if we have the DNA is perfectly intact and everything's just right, then the cell divides during the mitotic phase. So that's the cell cycle, right? So now let's go over mitosis. So mitosis contains 5 stages. Prophase. Sorry, 6 stages. Prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So these are the steps of mitosis. So let's go over the steps of mitosis then. So it's also important to note the difference between diploid and haploid. Oh, well, let's actually get into that later in meiosis. Right now, let's just concern ourselves with something. Let's concern ourselves with a cell of 2n equals 4. So now we get to talk about sister chromatids. So we have a cell in G2, right? Our cell looks like this ordinarily, right? It looks like a bowl of spaghetti. So this is G2 phase, right? Now after G2 phase, and we're going to mitotic phase, now we're starting prophase 1. Sorry, we're starting just regular prophase. So now during prophase, what's going to happen is that the DNA in the nucleus is going to condense. So now we get this more recognizable shape. Now they look like X's, right? So now I want to note, right? Look at this pair of chromosomes, right? They have the same size. And look at this pair of chromosomes. They also have the same size. So now this is where homologous chromosomes come into play. Homologous chromosomes. I think I spelled chromosomes wrong. I can never tell if it's with an A or with an O. It's with an O. Okay, so yeah, I was right. I always spell it wrong, so. Anyway, so homologous chromosomes. What homologous chromosomes are is that they're not quite identical, but homologous chromosomes always come in pairs, right? So these pairs are not identical. But what they do have in common is that they have the exact same size. Not exact same, but they have similar size and similar genetic information. Sorry. What? Why does it keep doing that? Okay, hold on. Okay, there. Finally. Jeez. Anyway, sorry, it's a little finicky. Anyway, so homologous chromosomes are of similar size and similar genetic information. So now what does this mean? It essentially just means that, well, it means just exactly that. They're similar size and similar genetic info. Now, you get, so this is where you get one from your dad, one from your mom. You have this one that came from dad, and this one that came from mom. And this is what makes you so special, right? Is because these two, this combination of chromosomes are very, very unique. And we'll get more into what this means later during meiosis. But essentially, what it means is that when we have 2n equals 4, because we're a diploid organism, this is what being diploid means. It means to have homologous chromosomes. Homologous chromosomes mean that you have two chromosomes that are of similar size and similar genetic info. But it's also important to note that each one of these chromosomes, each one of these X's, are still one chromosome. Now, if we zoom in a little bit further, right? If we zoom in a little further, you'll see that this is kind of where our X's come into play, right? because you'll see that we have 4 sister chromatids per. Well, okay, no, that's confusing. Okay, we have 2 sister chromatids per 1 chromosome. Now, what are sister chromatids? Now, sister chromatids, excuse me, are identical size and identical info. So that's the difference between homologous chromosomes and sister chromatids. Now, homologous chromosomes, let's draw that out, would be like this or like this. Similar size, similar genetic information. But these within these duplicated chromosomes, so chromosomes would appear like this after the S phase of the cell cycle. These would be sister chromatids because sister chromatids are attached at the centromere. The center of the chromosome, after it's been duplicated, the center is known as the centromere. Right? So then you have each one of these sister chromatids, you have 2 for every 1 chromosome after it's been duplicated. Now, like I said, they're identical size and identical info. And because they are attached here at the centrosome, it would still be considered 1 chromosome. So homologous pairs, so this would still be a homologous pair, for example. That's 2 chromosomes. But there are 4 sister chromatids. But that doesn't mean there are 4 chromosomes. There are still only 2 chromosomes. And I know that's confusing. It just takes some getting used to, I suppose. But that's just what it is, right? 2 sister chromatids for 1 chromosome that's been duplicated, and then there are 2 chromosomes per homologous pair. I really hope that's clear because this is really important. I'm just gonna leave that actually. Okay, so after prophase, then we go into prometaphase. So during prometaphase, then these centrosomes start to actually attach themselves at the centromere. This is a little misleading, but bear with me. So this is prometaphase, right? During prometaphase, well, I guess I haven't really included this in any. Okay, if you'll excuse me for one moment, sorry. Okay, well, it's not really a detail that you need to know, but it's something that I still want to emphasize. Okay, so during prophase, you still have this nuclear envelope in the cell, right? And then during prometaphase, then it actually dissolves. It's no longer there. So, but like I said, it's not really a detail that you need to know. So I'm not really going to include it. Oh, sorry. But anyway, during prometaphase, so the nuclear envelope has dissolved, and then these. So let's actually just draw it in. Why not? So the nuclear envelope has dissolved, and now the centrosomes have attached at the centromere, and then this is prometaphase because it's like a transitional period between metaphase and prophase. So now we're going to go into metaphase. Everyone forgets about prometaphase, by the way. I do too from time to time. So now during metaphase, each one of these chromosomes are now going to line up at the center. And they're going to line up via the microtubules, or aka mitotic spindles, from the centrosome. They're going to just attach here at the center, right? So now this is metaphase. So now after metaphase, then we have anaphase. So what happens during anaphase? Now during anaphase, this is really important, right? I mean, it's all really important, but sorry, just let me get a nice cell shape. Okay. So now during anaphase, then the sister chromatids separate out. So what does this mean for the cell? It now means that these sister chromatids would now be considered chromosomes because they're now separated, right? Because these are equal, these are identical copies of one another, these sister chromatids. If anything, they should be called twin chromatids, but anyway, I'm not a scientist. So now this is anaphase. So during anaphase, so after metaphase, when everything's lined up, anaphase is when the sister chromatids actually start to split apart. And now we finally get into telophase. So now telophase. I'm sorry, I'm kind of running low on space here. Actually, let's go ahead and erase this. Sorry if you needed that, but you just go ahead and rewind. So during telophase, we have what's known as a cleavage furrow that's now being formed. So also, the mitotic spindles start to dissolve a little bit. They start to go away because they're no longer needed. And now during telophase, so we had our DNA that was condensed, right, in the X form and now in this line form, right? We have now the DNA that's going to start to decondense. So now it's going to start looking like the bowl of spaghetti. Well, that's actually no. Now it's going to start looking like the bowl of spaghetti again. But notice how I'm drawing 4 distinct lines here. Because if we split this in half, now each cell has 4 chromosomes. So we have exactly the amount of genetic information as we started with. We have still 4 chromosomes. So this is telophase. And then after telophase is cytokinesis, right? Now, and then cytokinesis, we have one here, and then, I don't know, another one here. Oh, by the way, the nuclear envelope starts to reform. So that's where the nucleus is. So then we have the nucleus. With all the DNA in it. It kind of looks like a... never mind. So now we have 2 daughter cells, both of which are exactly equal to what we started with. Have the same genetic information of... sorry, they have the same... Okay, so sorry, here we go. So now both of these cells are now 2n equals 4. Just like we started with. 2n equals 4. So now this was mitosis. Yeah, now we're going to go ahead and move on to meiosis. So now meiosis is where there's quite a lot of confusion. Because this is where we start to really go into the difference between sister chromatids versus homologous chromosomes, and then there's just a lot of misconception on the way. So we can just go ahead and move right on, and I'm gonna take it a little slow as well because I understand this is quite difficult. Well, I don't want to take it too slow because I've been here for a while, but anyway. So now we're going to go over meiosis. So meiosis has 2 phases: meiosis 1 and meiosis 2. Now meiosis occurs after G1, and then after S phase, and then after G2. So it's important to note this because after S phase, we still have the DNA that's been duplicated, right? And then we go into meiosis. So the cell is going to receive signals to start producing gametes, and it's going to do this through meiosis. So what is a gamete, right? A gamete is a sex cell. Now, what is a sex cell? A sex cell can include an egg, or it can include a sperm, and that's just about it. In humans, that's what it is anyway. And that's what it is for most animals. So egg and sperm, those are your gametes, or aka sex cells. So, and then if this is for your information as well, somatic cells are made during mitosis. Somatic cells just means body cells. So that's like every other cell in your body are somatic cells versus gamete cells, which are sex cells. So the point of G1, S, and G2. Sorry, no, that's not right. Sorry. So the point of meiosis is to make gametes, and we're going to do this basically pretty much in the same. Sorry, it's going to get initiated pretty much in the same way as just regular cell division. Okay, so we have just like a stem cell, right? A stem cell which is 2n equals 4. So this stem cell has this nuclear envelope and then 4 chromosomes in it. It looks like a bowl of spaghetti, right? But this stem cell, okay, also this is during G2, by the way. It's gotten signals to do meiosis instead of mitosis, right? Because the body is starting to produce gametes. So it gets a signal, and it's like, okay, you got the check to start doing meiosis. So now, instead of going to mitosis and splitting that way, it's going to do meiosis. So now we're going to start in meiosis 1. So meiosis 1. So there is, so just like kind of very similar to mitosis, meiosis 1 is in 5 steps this time. Prophase 1, metaphase 1, anaphase 1, and telophase 1. I did say 5 though, so we're also going to include cytokinesis. So that's meiosis 2. And very similar to meiosis 2, we have meiosis. Sorry, that's meiosis 1. Very similar to meiosis 1, we have meiosis 2. And I just realized I didn't include. There we go. Okay, so these are your phases of meiosis. So now let's just go ahead and go over it now. So, oh, whoops, didn't want to do that. Actually, I might have to expand it. Never mind. Actually, let's zoom in a little bit. Actually, right. So we start off with the cell in G2, and then now what the cell is going to do. So we just skip right over prometaphase, right? And we're just going to do everything in prophase. So there's going to be the attachment of mitotic spindles, and there's going to be the condensing of DNA, right? So this is so now we have. Okay, sorry, one sec. So like I said, this is 2n equals 4. Actually, better yet, how about this? Okay, and then we have the mitotic spindles starting to form. And then that's the cell. So now this is during, sorry, Prophase 1. Okay, so what's going on here, right? Now, as you saw with just regular mitosis, I made a pretty big. I made no distinction between the types between the chromosomes, but here I did make a distinction. Now here, red is from mom, and white is from dad. And now here is where that important difference comes into play, because after meiosis 1, these homologous chromosomes are going to actually split apart. They're no longer going to be together, right? Which is sad for mom and dad, but anyway, they're going to split apart. But also pay attention to what's going on here, right? So, and then we're going to go more into independent assortment during metaphase 1. Um, anyway, so look at this also. During regular prophase, they are side by side essentially. These homologous chromosomes. But during prophase 1, they're literally overlapping. So what is happening during this overlapping? This is what's known as crossing over. This is known as crossing over. What happens during crossing over? Genetic information is exchanged. This is incredibly important for genetic variation. The genetic info is being exchanged during crossing over. So you have different nucleotides that are being traded over here, and then different nucleotides that are being traded over here. And it all works out because this is natural. This is how genetics works. It's through this genetic variation that is why sexual reproduction is so very important, right? So that's what's happening here. The genetic information is being exchanged during crossing over. That's happening here at each intersection point, and it's happening here. So that's something that's very important about meiosis 1, and that's happening during prophase 1. So now we move on to metaphase 1. So there's another big distinction made between metaphase 1 and just regular metaphase, and that's this. Actually, let's draw our spindles first because we're gonna change it around a little bit. Actually, let's try a little longer. So during metaphase 1, there's a really big distinction that's being made here as well. Do you notice a difference? These chromosomes are now lined up horizontally instead of vertically. So instead of being one by one by one by one, right? Now they're one by one, one by one, one by one. So they're in pairs now. And it's also important that these aren't just any pairs; these are homologous chromosomes as well, right? This is a homologous pair. This is a homologous pair, right? That's very important to note. And also look at what's going on here. We have a red chromosome here, and then a red one here, and then a white one here, and then a white one here. But that can change. And that's a very important. It's also very important about meiosis 1 because we could have two reds on this side and then two whites on this side, or we could have two whites on this side and then two reds on this side. Any this is a variation, right? It's very important because now we have genetic variation coming into play again. Now, so this is called independent assortment. This is called independent assortment. I'm trying to find a good place for this. Okay. Independent assortment. So what is that? What is that? Chromosomes line up independently of each other, creating variation. Also, this is metaphase 1. Right? So that's like that's something really important to note, is that independent assortment, right? I guess it got kind of cut off, and it's fine. Anyway, sorry. So we have two very important genetic variation steps, and that's crossing over and that's independent assortment, right? So crossing over independent assortment, that's those are two very important aspects of meiosis 1. So after metaphase 1, then we're going to go into anaphase 1. So now during anaphase 1, now the homologous chromosomes are going to be separated from each other. And also notice here that in this case we're not separating sister chromatids, we're separating homologous chromosomes. So now here we could see the distinction, right? Now on this side we have two chromosomes, and then on this side we have two chromosomes as well. So now we're starting to make n equals 2, which we'll get into after meiosis 1 is done. So now this is anaphase 1. Right? That's when we're pulling apart homologous chromosomes. And then we go into telophase. And then we go into telophase 1. Now during telophase 1, unlike ordinary telophase, the genetic information, the DNA does not decondense. Right? So here we have a big X, then a little x, and then a little, and then a big one here. So look at the sizes of these. These were once homologous chromosomes, and these were once homologous chromosomes as well. But now, during telophase, we're actually splitting it apart. And then after telophase 1, then the cell splits apart. The cell splits apart to make, so this is cytokinesis, right? To make two cells, both of n equals 2. So what's the difference between n equals 2 and 2n equals 4? Well, the number is just denoting the amount of chromosomes. So now these gametes are what's known as haploid. So these are going to be haploid. And these ones are going to be diploid. So what does that mean? Diploid just essentially means that something has homologous chromosomes, right? It has homologous pairs because you get one from your dad and one from your mom, right? But haploid, then those homologous chromosomes are actually separated, right? They're actually going to be separated. So once they're separated, now it's n equals 2. There are no more homologous chromosomes. Because then you have 2n equals 4. Well, 2n equals 4 tells us that there are two pairs of homologous chromosomes, right? So two pairs of 2, that's 4, right? But n equals 2, that just tells us there are 2 chromosomes. There are no pairs. That's what that's telling us. So that's the difference between diploid and haploid, right? So now we can go ahead and go into meiosis 2. So I'm going to go ahead and erase this. So I apologize. Take screenshots if you need. So now meiosis 2 is pretty much exactly like regular mitosis. So let's draw our cells again. Well, I'm just gonna draw. Well, okay. Well, I'm gonna draw both cells, but we're gonna proceed with just one, right? Because then we have one big X, and then one little x, and then one little x, and then one big X. So let's go ahead and proceed with this one. Right, this is happening simultaneously to both cells, by the way. But I'm just using an example for one. So this cell just goes right into prophase 2, right? So after this is after cytokinesis. Now this cell is going to go into prophase 2. So if you notice, there's no genetic information to condense. There's no DNA to condense. So there will be no condensing the DNA during prophase 2. What's just going to happen is that the mitotic spindles will go ahead and start attaching. Right? So this is metaphase 2. Sorry, prophase 2. So after prophase 2, then what? So then there's metaphase 2, right? So then we have the so like I said, meiosis 2 is very similar to mitosis because now we're actually going to separate the sister chromatids. Because if you look here, they're still in the X conformation. They still look like X's. They're not quite like lines. They look like X's, right? So now this is metaphase 2. Right? So now they're going to line up one by one vertically, and now the sister chromatids are going to be separated. They're going to be pulled apart. So now, and that happens during anaphase. Anaphase 2. So now the DNA is no longer duplicated. The sister chromatids are separated. Right? That's all happening during anaphase 2. And now, during anaphase 2, these sister chromatids will now be considered their own chromosomes. So now this is anaphase 2. Okay, well now what? So now we're going to go into telophase 2. So telophase 2, we're going to continue to make this cleavage furrow, right, as we've seen many times before. And now in this step, now the DNAs are going to start to decondense. Right? And then we still have, and then the cell is still considered 2. Both of these cells are still considered n equals 2. Well, both of these preliminary cells anyway, they're still considered n equals 2. So that's telophase. That's telophase 2. Exactly how you'd expect. It's happening exactly how you'd expect because it's very similar to just regular telophase during mitosis. So now these are going to split apart and make 2 daughter cells. Right? And we're actually gonna make a total of 4. Remember, because this cell is going to split apart as well. So we have a total of 4 daughter cells made for meiosis. Okay, so I hope that's clear. That was meiosis and mitosis. But yeah, There's one more thing I want to go over. So we went a little bit over about genetic variation and whatnot. So let me show you why the distinction between homologous chromosomes is still really important when it comes to this. Right? So if I have a sex cell, right? Okay? So that's one sex cell. This is n equals 2. And then we have another sex cell, also n equals 2. Let's say this is from your dad, and this is from your mom. So your mom and your dad are going to go ahead and, fertilize an egg and whatnot, whatever euphemism you want to use. And what's going to happen is that these cells are going to, sorry, these cells are going to come together. The, the sperm is going to fertilize the egg. And notice how there's still both n equals 2, right? So the sperm is going to fertilize the egg. And now, after this has happened, we're going to create one single cell. So now the cell is going to look like this. So now this cell would be a somatic cell, and it would be 2n equals 4. So now we've created a diploid from two haploids. Two haploids from your dad and from your mom. Fertilize an egg to make this. And look here, these are still homologous chromosomes. You get one from your dad and one from your mom. You get one from your dad and one from your mom. That's what's happening here. And that's why fertilization is so important, because you still get the homologous chromosomes. They've just been split. They've just been separated from meiosis 1, because here you have the gametes that don't have homologous chromosomes, and now you have the somatic cells that do. So that's what's happening during fertilization. Okay, so let me just go over the questions that are left real quick. Um, if you'll bear with me for a second, I'm sorry. Um, let's see. Okay, we can go over this question. Okay. Okay, we can go over question number 24 and 25, because I've gone over pretty much everything else, and I don't really want to. I've gone over quite a long time, and I have other things to take care of. So if you'll excuse me for one moment. Um, okay, so question 24 is asking. Um, Question 24 is asking, what is a cleavage furrow? So we went over what a cleavage furrow was. I don't think I actually made it super clear. So during telophase, this is when a cleavage furrow is being made. It kind of looks like a butt. That's how I imagine it anyway. So then we have the DNA decondensing. This right here, that's the cleavage furrow. So when you say cleavage furrow, think of this. Anyway, so what this is just kind of like a vocab question, if anything. What is a cleavage furrow? A cleavage furrow is the pulling in of a plasma membrane between developing animal daughter cells. That's a cleavage furrow. It's not metaphase plate where the chromosomes attach to the spindle. That's during metaphase, of course. This is during telophase. A ring of vesicles that forms a cell plate in plant cells. Well, that's how plant cells divide, which we don't really talk about plant cells in this class. And then an indentation between daughter prokaryotic cells, which once again, um, this answer D implies that it's the actual splitting has occurred. But what the cleavage furrow is that it's a pulling in of the plasma membrane between developing outer cells. So when you see this, it's pulling in on itself. It's essentially just being pinched off into two different cells. So that's what a cleavage furrow is. And then again, once again, this is kind of a vocab question. A genome. I don't think we really went over what a genome is, but which of the following statements correctly describes an organism's genome? An organism's genome. Um, 1 second. Okay, so an organism's genome is going to be a genome is a complete set of all of an organism's genes. Um, I don't really have a good way of remembering this. Honestly, it confuses me a little bit too. But just know that an organism's genes, and mm, I don't want to say anything wrong actually. If this is just what a genome is, I don't. I would, if I were you, I would consult the book if you're still confused about it, because honestly, I'm still confused about it. But that's just what a genome is. It's not proteins. It's not a single portion of the DNA. It is the complete set of all of an organism's genes. Nor is it genes found in the gamete. It's B. B is the correct answer. So anyway, I've given you all of the tools to answer the rest of the questions because honestly, I'm getting quite tired. It's almost midnight. Um, but there is a few more things that I still need to go over. Um, so just give me 1 sec. Okay, so Dr. Howe has decided to add protein or to add cell signaling to your curriculum. So we can go over that very briefly. So when we're talking about cell signaling, we need to know what a kinase is. So a kinase is an enzyme that phosphorylates something. So that something can be literally anything. It could be sugar. It could be another enzyme. It could be a fat. Well, maybe not. It could be a protein. It could be DNA. It could be a whole laundry list of something. A kinase is just an enzyme that phosphorylates something. Now, how does it do this? It does this via ATP. So now let's look at the structure of ATP. If you remember your if you remember your sorry if you remember your nucleotides, you'll notice that ATP is actually adenosine triphosphate, and adenosine is one of your nucleotides. So this is ATP because ATP has three phosphates, right? Adenosine triphosphate is ATP. So this is ATP, right? So now what ATP does is that it has these high energy phosphate functional groups that are going to be released, and they're also going to release energy. They're going to release energy, and they're going to add a phosphate group to something. So that's what kinase does. It phosphorylates something by using ATP, and ATP offers the energy as well. So now, what does this look like? So you have kinase. Let's just draw our kinase, right? So kinase, we have our substrate. The substrate is going to come in. It's going to do a conformational change at the active site of the enzyme. So this is, of course, is your enzyme. E for enzyme, and then so it's EK. E for enzyme, K for kinase. Um, so now it's going to fit in the active site. So this is your active site, of course. This is where the actual magic happens. Magic, aka chemistry. This is where the magic happens. And then this, so we have an induced fit of a substrate here. The shape changes a little bit. And then so then what we're going to do is we're going to take ATP in this process and we're going to turn that into ADP. So now in this process we're going to take the one phosphate group lost from the adenosine triphosphate into the adenosine diphosphate. That one phosphate is going to go ahead and be added to the substrate. So now the substrate is going to evolve into the product, which is now, well, PR for product. Excuse me. And then the phosphate. So now this product has been phosphorylated. And like I said, this could be anything. This could be a sugar, this could be an enzyme, or a protein, or DNA. That's essentially what just phosphorylating something is. And it's a kinase that does this. And I also want to emphasize that is the purpose of kinase. That is the only type of enzyme that you have to know for now, is a kinase. Kinase phosphorylates something. Very, very, very important. So now what does phosphorylation do? Right? So now that we have this this product that's been phosphorylated. So let's say this substrate is actually a protein. The substrate is actually a protein. So now that this protein has been phosphorylated, let's say that this protein is also inactive in the unphosphorylated state. So it's inactive. So now, after it's been phosphorylated, now we have an active protein. So now the addition of this phosphate group, has actually changed the shape. So I guess we could actually, this is entirely accurate. So let's go ahead and let's make it like a I don't know, like a diamond. How about that? Or like an octagon. Let's make an octagon. So it literally changed, this phosphate group literally changes the shape of the product of what it came from. So this we have the substrate, this protein, right? This phosphate group is literally going to change the shape of the protein, right? So now that it's changed the shape, now this protein can go ahead and do chemistry. So now we could have other things come into it. We could have other substrates come into it and come out as products. All because it was phosphorylated. Because this phosphorylation actually activated the protein. So now it can go ahead and interact with other particles. So this is all from just adding one phosphate group, which is pretty fascinating in my opinion. But anyway, so those are kinases and then phosphorylation. Okay, now we can talk about lipid hormones and versus steroid hormones. So lipid hormones. Let's look at an example of a lipid hormone. Let's search up estrogen. Oh, that's not going to work. Okay, perfect. This is estrogen, and let's look up insulin as well. Whoa, okay, that's way too big. Okay, so this is insulin on the right, and this is estrogen on the left. So, what do you notice about them? So, insulin, of course, is a protein, and estrogen is a lipid. Estrogen was what's known as a steroid. Right? Which you should hopefully be familiar with steroids at this point, but it's a type of lipid. So, anyway, so these are both what are known as signalers, cell signalers. So when we're talking about hormones, not only are they signalers, but they're both hormones, right? So they both perform different functions within the cell. But how are they different, right? This one's a lipid, and this one's a protein, of course. But what does that mean? It means that the steroid is going to be nonpolar, and the protein is going to be polar. So what does this mean for the cell? It means that this one is that protein hormones will bind to receptors outside of the cell. So that means on the cell membrane and whatnot. And then the steroid hormones will bind to receptors inside of the cell. Okay, well, why is this? Well, this steroid, if you recall way back when I started, if you recall, the steroid is just going to go right through the lipid bilayer. It's going to go right through because it's nonpolar, right? It's despite the fact that it's still large, it's still going to go right through because it's nonpolar. How about protein hormones? Proteins and hormones are not going to go through because they're very large and very, very polar. So they're going to bind to receptors on the outside of the cell. So like peripheral membranes and whatnot, and then that sort of thing. So this one's going to go on inside, and this one's going to go on the outside. It's another thing that Dr. Howe wants to emphasize. And then lastly, we're going to go over the GPCR. What is GPCR? G protein coupled receptor. That's a mouthful. And it's a lot easier for me just to look it up and show you what a GPCR looks like. So what does this do? Okay. So this is your GPCR. So not pictured here is actually, whoops. Not pictured here is is um is a receptor site. So now, what's known as an agonist, or in this case, so the agonist is essentially what's acting as the cell signaler. So you have this site in which something can be bound. So now this is going to be bound, and it's going to do a conformation change. And now once this is bound, once this signal is bound, then GDP is going to be replaced with GTP, right? And if you notice, the G protein is essentially in two subunits. You have the alpha subunit, and then you have the beta gamma subunit, which you don't need to know the specifics. This beta gamma subunit is once this signal has come in, this beta gamma unit is going to release, and GTP is going to be bound, or rather, GTP is going to be bound and then it's released. But anyway, they go in tandem. The these two portions of the protein are going to separate after GTP has been attached. So that's really the only thing that Dr. Howe wants to emphasize for the GPCR, right? GDP, here's how I can remember. GDP, G protein is deactivated, right? And then vice versa for GTP. GTP, G protein is activated. So GDP, D for deactivated, GTP, you get the idea. Okay, so anyway, I'm done for now. It's been over 2 hours, so I'm a little tired. So yeah, I'm gonna go ahead and go to bed, and I hope you guys have a good night too. I hope you guys have a good exam and a good time on Friday, and then a good spring break as well. Because I know your exam is gonna go just fine if you hopefully watch this video. If not, I really hope you skip to the parts which were important and whatnot. So like like the organelle review and then the cell cycle review. So yes, I thank you for watching, and I